Assalamualaikum warahmatullahi wabarakatuh and good afternoon to Dr. Nir Arhan. My name is Fairuz Mohd Salleh, and today I would like to present my research project on essential oil production from Persicaria odorata leaf of effect of sample size and extraction time, supervised by TS Dr. Azdarina Yahya. And this is overview of my presentation today. First, I will go through the introduction, then problem statement, objectives, sample preparation, extraction process, and analysis. Okay, first, I would like to do the introduction first. Our main focus, which is essential oil, is a mixture of bioactive compound extracted from plants, which are characterized with a highly concentrated aroma or scent. So this compound is categorized as secondary metabolites because essential oil do not involve in the plant growth but instead serve other functions such as defense, reproduction of the plants, and communication between plants. Essential oils always have been part of medical practice of ancient times due to the natural remedies of source plant of the essential oils. So the history of essential oils spans thousands of years across different civilizations around the world, from the western region to the eastern regions. So for example, the scent of essential oil has been recorded to stimulate the body and mind to be more relaxing, improve sleep patterns, and promote mood enhancements. However, the reliability of ancient texts as direct source is still limited and requires scientific research to verify the claims. So this is fun fact that essential oil practice is dates over back to 5,000 more than 5,000 years ago. It's first used by the Greeks, Romans, and Egyptians, and has evolved alongside the human civilization until today. And next, I would like to do the problem statement. So, why I use the sample of Persicaria odorata? Persicaria odorata, or short as P. odorata, is a species of flowering plant that grow in the tropical regions. So this plant is commonly known as Vietnamese coriander in English, daun kesum in Malaysia, Phak Pai in Thailand, and Raram in Vietnam. So, P. odorata has a distinct scent which indicates the presence of essential oil. So, the scent of the P. odorata has been described as has a coriander-like aroma with a pungency and spiciness. So why this plant? So why not the plant? Why this plant? So P. odorata has been claimed to have of traditional treatment for illnesses. So for example, the practice that claims P. odorata can act as natural relief against ailments such as fever, nausea, snake bites, muscle sore, ulcers, small wounds, and promote hair growth. So as I said before, uh, without scientific evidence, we cannot take this as a um, we cannot take that as at face value. So we need to do scientific verification. So in this case, for P. odorata, pharmacognostic studies has concluded that this claim may be useful. A research have done by Dash and Zakaria in 2016 has reported that P. odorata contains presence of steroids, terpenoids, flavonoids, tannins, and phenolic compounds, carbohydrate, mucilage, proteins, and amino acids. Uh, so that's in back in 2016. In 2020, uh, Rashidi Nejat and Jafari has cross-referenced the compounds in the P. odorata, and it claims that their claims that P. odorata has high potential in food, pharmacy, and agriculture and cosmetic industry. So the problem for these plants or extraction as a whole, the essential oil use typically around 5% of the plant mass use. So it's highly dependent on outside factors such as harvest seasons, climate, and plant condition. So, in this study, we want to decrease the gap between predicted yield and actual yield, so that when in industry, we can get the best yields as we can, the best yields as we can. So. During my literature review, I found the research gap that recent studies of Biodata is focused more on the bioinformatics and bioactivity capabilities of the plants. However, the studies that focus on different extraction time and sample size via hydrodistillation are limited. So basically, that's why I want to do this. I did this research project. So we want to study that focus on the extraction time and sample size. Now, from the research gap, we go to the objective, which is I have three objectives. First, the essential oil yields, morphology versus yields, and natural compound properties. So first, I want to study the effect of extraction times and sample size toward the essential oil protection yields from the P. odorata plants. Second, to analyze the effect of extraction time towards morphology of P. odorata plants after essential oil extraction. And third is to study the effect of extraction time and sample size towards antioxidant capability of essential oils from P. odorata plants. So now we'll go to the methodology. First the sample preparation. So the fresh plants is locally obtained at Pasar Besar Kelang Meru, and we have four steps of preparation. First is the segregation. The fresh sample is manually sorted to remove foreign matters and non-target plant species. So when we purchase the plant, sometimes there are other plant matters that is exist in the plants, such as grass, even small animals. So we need to separate them. Second is cutting, which is the fresh sample is cut into small pieces, not smaller, that is between 1 inches and 2 inches. Third is the drying. So the cut sample is cold dry in open space at minimum 3 days. After drying, we will collect them and we will store it in a dry and cold place. Then the extraction process, which is as said in the problem in the research gap, we will use the hydrodistillation process. This is due to the multiple advantages. The first is solvent-free method. Hydrodistillation does not require organic solvents, making it environmental friendly and safe use for food, cosmetic, and pharmaceutical application. Hydrodistillation can prevent the thermal degradation of the plant by immersing plant materials directly in boiling water. The temperature is naturally controlled around 100 degrees Celsius, which prevent the overheating and charring of the delicate essential oil. And third is the most important is the simplicity and cost effectiveness. Hydrodistillation is relatively straightforward and inexpensive technique compared to the modern methods. So this is the schematic diagram of hydrodistillation. So the plant sample will be put into the flask with water and will be heated by the flask heater. So the water will evaporate into steam and carry the essential oil volatile compound, and will go into the top chamber and we will cool down by refrigerant. The refrigerant used will be the tap water, and then after that it will condense and fall into the Dean stack, where the volatile organic compound will naturally separated from the water due to the polar and non-polar properties. Once the extraction is done, we will decant them using decanter over here. Now we will go to straight to the analysis. First, the antioxidant activity evaluation. So why evaluation on the antioxidant is matters? Antioxidant evaluation confirms that the essential oils correspond to the preserved biological activity, not just increased quantity. So basically, this is just we want to preserve the quality of the oil. It will be useless if we had the high yield but the quality is low. We have two antioxidant evaluation, which is first is the DPPH assay, which is a simple spectrophotometric method to measure the substance antioxidant power by assessing its ability to scavenge the stable DPPH radical. It will reduce the deep violet radical to pale yellow non-radical form. And the second one is the FRAP assay, which is a fast and impressive method to measure the total antioxidant capacity. It will be detected by the ability to reduce ferric iron to ferrous iron, creating deep blue color chains. So these two evolution will be carried out on the Polar Star Omega microplate reader, and it will compare to the ascorbic acids antioxidant activity graph. Second one is bioactive compound identification. So why this is matter to ensure that the essential oils correspond to the chemical quality and biological effectiveness rather than quantity alone. So basically, this is the same with anti with the antioxidant capabilities. We want to preserve quality. So how we want to identify the bioactive compound? We will use two analysis equipment. First is the GCMS, the gas chromatography mass spectrophotometer, an analytical equipment that combines two instruments to separate, identify, quantify component in complex chemical mixture, ideal for volatile compound. And second is the LCMS, which is liquid chromatography mass spectrophotometer. So LCMS is work identically with GCMS, with one difference is it works best for non-volatile and thermally unstable compounds. And the third one is functional group identification. So this is all uh, uh, almost the same with the two, which is with antioxidant evaluation and the biotic compound evaluation, which is also we want to preserve the quality. So functional group identification, we will use FTIR, which is to identify the functional group by measuring how the absorbed infrared light frequency is rated unit fingerprint for each group. So each group, for example, alcohol, we have alkane, alkenes, terpenoids, basically any chemical groups that can be measured by FTIR. And the fourth is the morphological characteristic. So why this matters? We want to visualize the structural change in leaf tissues to explain the essential oil release mechanism. So we will use the SEM, which is um, SEM is an instrument that creates highly magnified image of sample surface by scanning it with a focused beam of electrons, revealing detailed topography and composition at the micro and nanoscale. And that is we have I have three conclusions for plant sample extraction process analysis. So for the plant samples, P. odorata or Vietnamese coriander or Donkosum has history of traditional claims of illness treatment. Combined with pharmacognostic studies that support these claims and variable research gap, this material currently serves as one of the best samples to academically focus on. For extraction process, hydrodistillation is an effective and sustainable extraction method, preserving volatile compounds by preventing thermal degradation. Hydrodistillation process is easy to maintain, a good starting point is in extraction studies. And finally, analysis. Chemical functionality, biotic compounds, and antioxidant activity may vary with extraction conditions. An integrated multi-analytical approach is essential to fully evaluate essential oil quality, linking yields, chemistry, bioactivity, and morphology of the samples. And this is the reference that I used to write these presentation slides. And that's all for me. Thank you.